Hello everyone, I welcome you all to Biology Lessons with Kalyani. In today's class, we will be discussing about enzymes. So this particular session is an introductory class. There will be a part 2 of this lecture series about enzymes where we will discuss in detail about the mechanism of action of enzymes. So let's check out what's our outline today. So first we are going to discuss what are enzymes, and second we will see how enzymes differ with respect to the chemical catalyst. Then we will have a broad overview about the biochemical reactions and how enzymes influence the biochemical reactions. Finally, we will see how enzymes are classified today and what is the nomenclature which is used for the enzymes in these days. So let's first see what are enzymes. Enzymes are biological catalysts which accelerate the rate of biochemical reactions. So enzymes are catalysts. Catalysts are substances which accelerate the chemical reactions. As in case of enzymes, biological reactions are involved and the rate of biological reactions is increased. So enzymes are called as biological catalysts or simply biocatalysts. Now these enzymes, if we see, what are they made up of? Basically, if we say, mostly all the enzymes are proteinaceous in nature. That means they are made up of polypeptides. But there is an exception. Some enzymes, they are made up of RNA. Example of it is ribozyme. So if you want to know a little bit about ribozymes, ribozymes catalyze reactions in which they split a long segment of RNA into smaller fragments, right? So all enzymes are proteinaceous in nature with the exception of ribozyme, which is an RNA, right? Now, as I told, enzymes are proteinaceous, so obviously it will have a primary structure, it will have a secondary structure as well as a tertiary structure. Now, this tertiary structure is responsible for its functionality. Now, if sometimes there are more than one subunit present, then quaternary structure can also be possible. Now, look. In case of the tertiary structure of the enzyme, there are many crevices or pockets that are formed, and these pockets are called as active site. Look, this is the image. This is the image of the enzyme. The enzyme taken here is chymotrypsin. What you should focus is that inside the enzyme, you can see the part marked in red. It is a kind of the pocket, and that is called as active site. So what happens in this active site? An active site of an enzyme is a crevice or a pocket into which the substrate fits. So in this active site, the substrate will be binding to the enzyme, right? Now let's see if we compare enzymes to that of the inorganic catalysts. What is the difference between the both? All the enzymes, as we already discussed, they are proteinaceous in nature, while the inorganic catalysts, they are made up of a combination of simple ions. So they are basically chemical compounds. Now enzymes, the reactions they catalyze, that is very specific with respect to the substrate, and they are the typically biological reactions, while that means the enzymes catalyze the reactions inside the living organisms, while inorganic catalysts, they catalyze diverse non-biological reaction. Clear? If we talk about the efficiency of enzymes, it is very high, while inorganic catalysts, the efficiency is less as compared to the enzymes. Now, the optimal temperature and the pH in which enzymes work is mild, while the catalyst, they can operate in extreme temperature and pressure. We can also regulate the enzymes by feedback mechanisms, but if we take inorganic catalysts, they cannot be regulated. So these are the differences between enzymes and inorganic catalysts. I told that enzymes always have a moderate temperature and pH in which they can work, but we have some organisms which are thermophilic in nature. What is thermophilic? That is the organisms which can thrive at high temperature. For example, in hot vents, in sulfur springs, volcanoes. So the organisms which are isolated from such extreme climate, they possess enzymes which show higher thermal stability. What does it mean? That the enzymes, they do not denature even if the temperature is as high as 80 to 90 degrees Celsius. Now, one of the examples of such enzyme is Taq polymerase, which is isolated from Thermus aquaticus. And this particular Taq polymerase enzyme is very essential because it is widely used in PCR for DNA amplification. So, thermal stability of the enzymes is extremely important. In order to understand how the enzymes, they catalyze biochemical reactions, we should first understand what are the type of changes a chemical compound can undergo. So a chemical compound can either undergo a physical change, which can involve either a change in the shape or the change in the state of matter, or else the compound can undergo the breaking of some bonds and formation of new bonds, which is called as a chemical change. So if we talk about chemical change, that means the bonds, few of the bonds are broken and new bonds are formed. Now, we can look at some of the chemical reactions such as your hydrolysis of the starch into glucose. That's an example of chemical reaction. And this is your conversion of barium hydroxide. That is also an example of your chemical reaction. So this chemical reaction, the rate at which the physical or chemical process is taking place, we can represent it as amount of product that is formed per unit time. So what is the rate of a chemical reaction? How much product is formed and in whatever time it takes to make that product. So rate is equal to amount of product divided by time. If we compare a reaction which is catalyzed by the enzyme to that of an uncatalyzed reaction where the enzyme is absent, then the rate of enzyme catalyzed reaction is vastly higher than the uncatalyzed reaction. So let's take the example of carbonic acid formation here. Now CO2 plus H2O, they combine together to form H2CO3 or carbonic acid. Now, if this reaction, it takes place when the enzyme is absent, then about 200 molecules of H2CO3 are formed per hour. But if enzyme carbonic anhydrase is present, then about 600,000 molecules of H2CO3 are formed, that too per second. So see, the reaction rate increases by about 10 million times. So enzymes act by accelerating the reaction rate. Let us now look into detail how the enzyme is able to catalyze a biochemical reaction in such high rate. So this is your enzyme and this is the substrate. The substrate is always getting converted to product. And what are the steps which are involved? So this substrate will diffuse and it will go near the enzyme and it will bind to its active site. So an enzyme substrate complex will be formed. This is a temporary state. The enzyme will act upon the substrate and it will break the old bonds, new bonds will be formed and product will be formed which will be still attached to the enzyme. That is called as enzyme product state. That state is also transient. Finally, what will happen? Enzyme and product will be released. The enzyme is now free to be used in the next reaction. Let us now graphically represent the same. So let's take x-axis and y-axis. Now this is your x-axis which shows the progress of reaction. What does it mean? How the substrate is getting transformed into product, right? Now let's see what's in y-axis. That's a potential energy. Now this is your substrate. This is your product. So this is the potential energy of the substrate, which I can represent it by A, and this is B, that is the potential energy of the product. The red curve, it shows the biochemical reaction without enzyme, and the pink curve, it is the biochemical reaction with the help of the enzyme. So first let's see just A and B. So what can you conclude from it? If we compare the potential energy of the substrate and the product, which is higher, we can see that the potential energy of the substrate is higher than the product. So we do not need higher energy for the substrate to be converted into product. So energy is also, it has to be released in this process. So this is an example of your exothermic reaction, because we know if in a chemical reaction energy is liberated as heat, that is called as your exothermic reaction. So next let's see what the graph for endothermic reaction. So this is your potential energy, this is the progress of reaction. If you see, this is your substrate and this is your product. Now if you compare the potential energy of the substrate and the product, the potential energy of product is higher than substrate. So what does it mean? It means that we have to give more energy for the substrate to reach the potential energy of the product. So this kind of reaction is called as your endothermic reaction. Is that clear? Now that we have understood how the graph will look for an exothermic and an endothermic reaction, we'll see a little more about energy. Now last slide we discussed about two types of reaction, exothermic and endothermic. Whatever type of reaction we are taking, immaterial of that, if you see, I'm using the same curve here. This is your potential energy, this is the progress of the reaction, and this is enzyme uncatalyzed reaction in black, and this is your enzyme catalyzed reaction in green. Now pay attention here. This is the substrate and this is the product. Irrespective of the type of the reaction we are taking, the substrate needs to have some energy in order to reach a very high energy transition state in order for the product to be formed. So whatever energy the substrate needs to have to reach that high energy transition state, that energy is called as the activation energy. Clear? If you see the black curve, which is without enzyme, the activation energy which you need is represented by this black line. And if you take enzyme catalyzed reaction, this activation energy is the difference between these 2 points, that is represented by your green line. If you compare the both, the activation energy required for enzyme catalyzed reaction is always very less. That's why the substrate needs very less energy in order to reach the high energy transition state in case of an enzyme catalyzed reaction, and that is how enzymes are able to increase the reaction rate by decreasing the activation energy. This is a very important concept, and if you pay attention, it's very simple. Okay, few more things in this. Now, enzymes, they can catalyze multi-step chemical reaction. So if you see, this is a multi-step chemical reaction, and that multi-step chemical reactions are always involved in a metabolic pathway. So we can take the example of conversion of glucose to pyruvic acid. Some 10 steps are involved in this, and 10 enzymes are also involved in this. Now, different enzymes can be involved in a metabolic pathway, or enzymes related to each other in an enzyme complex may also take part in the metabolic pathway. Another thing to pay attention is enzymes can catalyze different product in different conditions. Let's take an example here. Now, glucose, if you take, that is a substrate. Now, under normal conditions, glucose will be converted into pyruvate. But if you change the condition, that is if you make anaerobic condition, then lactate is formed, and this we can observe in skeletal muscles. Now, if you take example of yeast, the same glucose is converted into ethanol. So, different type of conditions may lead to the formation of different products. The final part is your classification and nomenclature of enzymes. We already had an introductory overview about enzymes. So now let's see how enzymes are classified. Typically, enzymes are divided into 6 classes, each with 4 to 13 subclasses, and we name the enzymes by a 4-digit number. You don't need to know a lot about the details about it, but I have just mentioned for your reference that this is how the enzymes are named. So the enzymes are classified on the basis of the types of reactions they catalyze. So they are of 6 types. The first is your oxidoreductase, second is transferase, third is hydrolase, L is liases, I is isomerase, and then ligases. So you can remember this using the mnemonic OTH Lily G. So you have 2 L, so you can just remember it by remembering the G for ligases. So that is how you should remember the name of the enzymes. Here EC1, the first one refers to the type of enzymes. Okay, rest you will be reading it in higher grades. Now let's now see what are the reactions that are catalyzed by the specific type of enzymes. Now if you take oxidoreductases or dehydrogenases, these enzymes are able to catalyze oxidation and reduction between the two substrates. So you have the two substrates S and S dash. So one of the substrate will be reduced, another will be oxidized. So example of it is alcohol dehydrogenase. Second type of enzymes is transferases, which are helpful in catalyzing transfer of a group. For example, this substrate has a group that needs to be transferred to this another substrate S dash. So transferases will be catalyzing such reaction. Sg will be converted to S, and the S dash will be getting the transferred group. Example is hexokinase, which transfers phosphate group to glucose. Let's take example of hydrolases. Hydrolases are enzymes that catalyze hydrolysis of either the ester, ether, peptide, glycosidic, CC, C halide, and phosphorus nitrogen bond. So example of it can be lipases and proteases. Lipases are responsible for lipid hydrolysis and proteases for the protein hydrolysis. Next category of enzyme is liases. So what do? Typically liases, they are responsible for removal of the groups from the substrates by some mechanism other than hydrolysis, and when liases are involved in a chemical reaction, they lead to the formation of the double bond. So take this example CXCY. Now we need to remove this X and Y. So what will be formed? X and Y will be removed and a double bond will be formed between the two carbon. Example of liases are pectin liase, pyruvate decarboxylase. So see, decarboxylase, it is removing the carbonyl group. So this is an example of your liases. Next type of enzyme is isomerases. As the name suggests, these are the enzymes that are responsible for catalyzing interconversion of the optical, geometric or the positional isomers. For example, if you take D-glucose, isomerases convert D-glucose to D-galactose. Similarly, glucose 6-phosphate, if you want to convert it to fructose 6-phosphate, that's also possible with the help of isomerase. Final type of enzyme is ligases. So what do ligases do? That these are the enzymes that link together two compounds. Okay, they typically form new bonds between the two compounds, and example of it can be DNA ligase. Now what does it do? It joins the two DNA fragments. So this is very widely used in biotechnological experiments. So it's a very important enzyme. So that's all about the enzyme classification. I hope you enjoyed the session about enzymes today. So we had a broad overview about what are enzymes, what are active sites, how enzymes act, their mechanism of action, how we are able to classify enzymes, and what is the mechanism behind enzymes catalyzing reaction at such a high rate. So please refer to the part 2 of the video, in which we will be covering the mechanism of enzyme action in more detail. That's all for today. Thank you everyone. Have a very good day.